Welcome to this educational video on gene regulation. Today, we will be exploring the different mechanisms that control gene expression in bacteria and eukaryotes. First, let's talk about gene regulation in bacteria. Bacteria are unicellular organisms that have a simple genome with a limited number of genes. They rely on gene regulation to ensure the expression of the right genes at the right time. Bacteria use a variety of mechanisms to control gene expression, including transcriptional regulation, post-transcriptional regulation, and translational regulation. Transcriptional regulation is the most common mechanism used by bacteria to control gene expression. In this mechanism, regulatory proteins bind to DNA to control the initiation of transcription. The most common regulatory proteins are transcription factors, which bind to specific DNA sequences and either enhance or inhibit transcription. Post-transcriptional regulation is another mechanism used by bacteria to control gene expression. This mechanism involves the processing of mRNA after transcription, which can affect the stability and translation of mRNA. Bacteria can also use small RNA molecules to regulate gene expression by binding to mRNA and affecting its stability and translation. Translational regulation is the least common mechanism used by bacteria to control gene expression. In this mechanism, regulatory proteins bind to mRNA to control its translation into protein. Now, let's move on to gene regulation in eukaryotes. Eukaryotes have a more complex genome than bacteria, with many more genes and multiple levels of gene regulation. Eukaryotes use many of the same mechanisms as bacteria, but with some additional mechanisms unique to eukaryotes. Transcriptional regulation is still the most common mechanism used by eukaryotes to control gene expression. Eukaryotes use transcription factors to control the initiation of transcription, but they also use chromatin structure to regulate gene expression. Chromatin is the complex of DNA and histone proteins that make up chromosomes, and its structure can be modified to either enhance or inhibit transcription. Post-transcriptional regulation is also important in eukaryotes, but it involves more complex mechanisms than in bacteria. Eukaryotes use RNA processing, including splicing and polyadenylation, to regulate gene expression. They also use miRNA and siRNA molecules to regulate gene expression by binding to mRNA and affecting its stability and translation. In conclusion, gene regulation is a complex and important mechanism that allows cells to control gene expression and ensure the right genes are expressed at the right time. Both bacteria and eukaryotes use a variety of mechanisms to regulate gene expression, with eukaryotes having more complex mechanisms due to their more complex genome. Thank you for watching this educational video on gene regulation.